Bismillahirrahmanirrahim. We start now the specimen paper 4, which has been now sent to us by Cambridge for the 2022 syllabus. And as you can see in this, there's 100 marks. This was is in the previous exams as well. But the only thing which is different is that the 15 marks section B has been removed. So now there is no choice. This is the first time that there will be no choice in the paper 4. Previously in the section B, there used to be a choice. Now there is no choice. And as you can see here, this is again 2 marks, but there's answer all the questions. There's no choice here. There used to be a section A and a section B, but now there is no section B. So there is no section B, and you will have to do all the questions. So there is no choice left anymore now in the paper 4. Now let's start with question number 1, AA. The A part of it, the II, whatever Dubentonia madagascaniensis, is a mammal native to Madagascar. IIs are active at night and make their nests high up in the trees. So they've given you some information. Active at night, make their nest high up in the trees. They feed on insect larvae in the trunks of trees. Figure 1.1 shows an II. The International Union for Conservation of Nature, IUCN, categorizes the II as endangered. This means that the II has a very high risk of becoming extinct in the wild. Name the domain to which the II belongs. Now you know domains are only 3: Archaea, Prokarya, and Eukarya. So this is because it's an animal, so it has to be eukarya. It can't be anything else because domains are only 3. The first, that is, we categorize all living organisms into 3 domains. State one reason why the II may have become endangered. Well, there's some things which you can write in every answer. Number 1 is habitat destruction. Maybe their habitat where they lived and where they breed and where they eat, that is destroyed. So maybe habitat fragmentation, maybe deforestation, maybe logging, and people have the urbanization, so they've cut down the trees where they used to live, because it says that I has a very high risk. And of course it tells you that it is night and make their nest high up in trees. So that was the question, and the information given in the question must be very carefully read and very carefully understood. So habitat destruction, maybe disease. Maybe a certain disease is rampant and that caused many of them to die suddenly. Or they're hunted for something. Maybe there's something, they have some. But here the ayah, of course, was considered very, because it had a very weird sort of a finger and would point. And people thought that if it pointed towards them, they would die. So they used to kill the ayah. It was sort of considered a bad omen, like in Urdu we say manhus hai. So they would kill it. So it was hunted or killed. So that is why it has become now endangered. State ways in which zoos may be able to protect this species from extinction. When they said it is zoos, so in zoo you do captive breeding. That means you keep the male and the female and you mate them and then you breed them and then you wait for the babies to be born, and then of course you release those into the wild. So captive breeding, number 1, release into the wild, number 2. Number 3, conservation projects in which people would be educated, education, raise awareness, and people would not cut down the trees where they were living, so their habitat was not destroyed or the food which was available to them was not removed. And research would go into them and their habits and how they could be, how their population could somehow be made to increase so that they breed and they are not disturbed. And veterinary care is that when they are sort of when they are in their time when they are breeding, that nobody sort of harms them in any way, so that then we can look after the young and maybe keep them in some protected environment and then release them into the wild. So these are general answers which you can nearly write in every question on this under this heading. Then coming to the B part of the question, there are 2 main II populations on the island of Madagascar, one in the west and one in the east. Figure 1.2 is a map of Madagascar showing the location of the 2 main populations. So there is this river flowing, you can see this. They've shown you this river flowing here. And then they've shown you the west population, which is this side, and then the river, and then there's a mountain range, which is this one here. The mountain range is this one here. And then there is an east population, and then there's another river here. So 2 rivers, mountain range, and then a west population, and then an east population. So look at this very carefully, and then it says a study into the variation in the DNA nucleotide sequence of IIs showed that there is a large genetic difference between the west population and east population. The two populations of II may be evolving into separate species. May be evolving into separate species. Now, here is a clue: east population, west population, one on one side, one on the other side, geographical isolation. Now, let's look at the question now. Now, the question is, with reference to figure 1.2, suggest why there is a large genetic difference between the two populations. And then it says, name the type of speciation that is most likely to occur. So you have to really read the question carefully. And as you know, this type of speciation, in which is geographical isolation, that has to be allopateric speciation. So that is a name which you must know, and so you got your 1 mark for that. Now coming back to this part, with reference to figure 1.2, suggest why there is a large genetic difference between the two populations. Now, basically, it's the same mark scheme points: geographical isolation, or you can say geographical barrier. You can see maybe that mountains or the rivers are a barrier. Then populations are unable to breed, so there's no gene flow. Then there are different selection pressures, meaning that maybe the environment on this side was less harsh. The environment on this side was harsh. There were too many rains. And then, of course, the population, the growth of plants would be affected. The trees would be affected. Maybe there's too windy conditions and the trees were all destroyed. So different selection pressures means different environmental conditions on the west side and on the east side. Then different mutations would occur. So different alleles would be selected for in the west and the east population. And this could be because of genetic drift, because a small number of them sort of separating and then growing on the population increasing on its own. Then separated for a long time. The west and the east population was separated for a long time. So there would be a change in the gene pool. So these are all the points of the mark scheme which I have just read out to you. And this is for you to be understanding it, how this west and east population had different selection pressures, large genetic difference between the two populations. Now I want you to pause the video here and I want you to memorize these words. And these are very typical words: geographical barrier. What was the geographical barrier? Rivers and mountains, which I've just talked to you about at the beginning of the question. Then the west and east populations were unable to breed. Different selection pressures, or if you said different environmental conditions. Then different mutations occurred. Then different alleles were selected which were advantageous, maybe the color of the fur or maybe the length of the tail or something which was advantageous would be selected for. Genetic drift may have occurred. Please everybody revise genetic drift. West and east populations separated for a long time. And the last point would be change in the gene pool. So you see, you must learn these points. You must learn the English, how to write these. Some of you, maybe know it, but you don't know how to word it. So you must be knowing to how to word this, that how, like these points, like different selection pressures, you can write every time. You can write mutations. You can write different alleles selected for. Genetic drift may have occurred. The populations are separated for a long time, so this will result in a change in the gene pool. So for 5 marks, these 5, 6 points, you should just memorize them. And remember then, whenever there is a question on it, that this is the wordings that you are going to use. The wordings are very important for you to memorize. Now, coming to question number 2, figure 2.1 shows a diagram of a motor neuron, the synaptic knob, dendrite, nucleus, Schwann cell A, B, and C. Now, what was A? A is the node of Ranvier. Now, everybody knows that. That is something which we all have studied, and I'm sure everybody knows that. So, node of Ranvier. Then, what was B? B is the axon. Axon, which carries impulses A or axon A away. And then C is the cell body. Wherever the nucleus is present, that is called the cell body of the neuron. So we know all this and we've studied this, so nothing to. Then it says describe the function of a motor neuron. Now, what is the function of a motor neuron? Basically, what does it do? It transmits impulses. So transmit impulses. You can't say messages. It says transmit impulses. These are electrical impulses. Or you can say action potentials. To where? To where? Motor neuron carries it to effectors. I always say, okay, easy to remember. M motor, M to muscle, or glands. There's a car called MG. I remember it from that. So effectors, or if you said muscles, or if you said glands, everything was correct. All these three were correct. But if you write something wrong and then you write the correct one, I'm sorry, that's going to be a +1 and a -1. So if you wrote muscles and then you wrote something totally different, like the lungs or something like that, well, that would be +1, -1. So there is always a +1, -1 if you negate yourselves. If you say something which is totally rubbish and one is correct and then the next you write something which is totally wrong, like if the answer was xylem and you wrote xylem and phloem, then of course xylem is +1 and phloem is -1. So that's the only place where we give you a -1. Then coming to the next part, with reference to figure 2.1, explain the fast transmission of impulses along a motor neuron. When it says with reference to figure 2.1, then you need to be looking at this. You need to be looking at this diagram. You need to be looking at this diagram, so then you know that what are we going to be talking about. Now, basically, what is the answer to that? The answer to that is very simple. The Schwann cells wrap around the axon. So the Schwann cells wrap around the axon. This is the Schwann cell, and it wraps around the axon to form the myelin sheath. So this is the myelin sheath, and then this insulates the axon. So the action potentials can only occur at the nodes of Ranvier, here, at the nodes of Ranvier, and local circuits form, and then it jumps from node to node like this. So it jumps from node to node, and this is of course called, the wordings for that is saltatory conduction. So just a quick recap of all what I just said. Schwann cells wrap around the axon to form the myelin sheath, insulates the axon. Action potential can only occur at the nodes of Ranvier, so local circuits form, and the action potentials jump from node to node, which is also called saltatory conduction. Question 3. Corals grow in shallow sea water. Corals consist of colonies of small animals called polyps. These polyps have photosynthetic protoplasts called algae within their cells, which is advantageous both to the coral polyps and to the algae. The algae that live within the cells of coral polyps can also live independently as free-living algae. The rate of photosynthesis of algae that live within the cells of coral polyps is higher. The rate of photosynthesis is higher than that of the free-living algae. Suggest and explain why the rate of photosynthesis in algae that live within the cells of coral polyps is higher than that free-living algae. No, basically the reason is there will be carbon dioxide concentration is higher. Why? Because that the coral or the polyps are respiring and producing carbon dioxide. So carbon dioxide is no longer a limiting factor. So more carbon dioxide means more Calvin cycle, which is the light independent reaction. And more CO2 means more of the binding of CO2 to Rubisco when RUBP and carbon dioxide combine in the Calvin cycle. Either you could have given me these points, or you could have said temperature would be higher. Temperature no longer the limiting factor because the coral and polyp is respiring and it has its own temperature. And more temperature means more Calvin cycle or more light independent reaction in the algae. And more temperature means enzyme catalyzed reactions are affected, which is of course the Rubisco enzyme, which is responsible for the Calvin cycle. So these are the reasons that you have to give me, is because it said corals consist of colonies of small animals called polyps. And inside these polyps, so small animals called polyps, and these polyps have photosynthetic protoplasts called algae within their cells. So animal cells have photosynthetic algae within their cells.
which is advantageous both for the coral and the algae. So you have to understand that what they were talking about. So the corals, these are the corals, and then the corals have animals called polyps, and then these polyps have photosynthetic algae inside them. So please first understand the question, read it 2 or 3 times, and then understand the question, and then think of how you're going to answer this and how you're going to come up with the answer. So suggest and explain why the rate of photosynthesis in the algae that live within the cells of coral is higher than that of the free living algae. So just to recap, CO2 concentration is higher, CO2 concentration no longer limiting, CO2 from the coral or the polyp is going to be respiring and producing carbon dioxide. Carbon dioxide is needed for the part of photosynthesis for which it is needed is the Calvin cycle, and CO2 binds to rubisco, and CO2 binds to rubisco and to RUBP. So how the Calvin cycle is going to be working out? So carbon dioxide plus rubisco gives you a 6-carbon intermediate, which immediately breaks up into 3 carbon and which is called GP, and then GP to TP, and that whole story of the Calvin cycle. Now coming to the B part of the question. Now the B part of the question now is a table. So the algae that live within the cells of coral polyps have 5 different chloroplast pigments. Table 3.1 shows the light wavelengths at which each algal chloroplast pigment shows its 2 largest peaks of light absorption. So chloroplast pigment, chlorophyll A, is at 430 peak 1 and it's 662. Then pyridinine is 456 and 485. Then 450 and 396 chlorophyll C2. Then dinoxanthin 442 and 471, and beta carotene of 454 and 480 nanometers wavelength. So look at the units. Corals can be kept in glass tanks that are usually lit by lamps radiating mainly violet and blue light with wavelengths in the range of 400 to 490 nanometers. Read it very carefully, 400 to 490 nanometers with reference to table 3.1. So keep on looking at this table. With reference to table 3.1, suggest why lamps radiating mainly violet and blue light are expected to increase the growth of coral polyps more than lamps radiating light of all wavelengths. Now you've got to see which ones are going to come between 400 to 490 nanometers, and why are they asking you this question, and this is for 3 marks. So you've got to figure this out in 3 marks, how you're going to answer this question as 400 to 490 nanometers. So you can see here now, as I marked these are the only 2 which were not in it. So 8 out of 10 pigments, 8 out of 10 absorb 400 to 490 nanometers. Only there were 2 which didn't absorb. So then you should have looked at this on this angle when you had read this question. Then photosynthesis of algae increases with more light absorbed. Yes, if more light is absorbed, naturally there's more photosynthesis. And this will of course transfer the oxygen to the polyps from the algae. And the coral growth will increase with more algal photosynthesis. So basically you have to correlate the respiration of one, the oxygen production of the other, and then of course the fact that the growth will increase with more algal photosynthesis, more oxygen available, more aerobic respiration. Now the last part of the question was a simple question. Photosynthesis in the algae living within the cells of coral polyps is the same as photosynthesis in plant cells. Outline the process of cyclic photophosphorylation. So cyclic photophosphorylation was something which is a direct question, and that's for 4 marks. Just a quick revision of the non-cyclic electron flow. You can see here, you've got photosystem 2 first, and then you've got photosystem 1. So electron excited to a higher energy level, accepted by the primary acceptor, then goes down to the electron transport chain and reaches here into the PS1, the photosystem 1. And then while this space is filled, another electron has already been excited from here by light. Light excites these electrons, and then of course when these electrons pass through the carriers here, then ATP is made, and we have ATP here being made, and this is called non-cyclic photophosphorylation. and then the electron from here passes to FD and then NADP reductase, and we have NADPH being formed. So 1 ATP, 1 NADH being formed. So we have this 6 times, and then of course 3 times we have another method which is called cyclic electron. So this is going to come back here and then it's going to come back here. Now you can see this in this diagram also. This is where the FS1, photosystem 1, PS1, sorry, electron is excited, doesn't go this side. No, it doesn't go this side. It goes back here. And then it comes back here, passes through the electron transport chain and comes back here, and ATP is made. And this is called cyclic photophosphorylation. So just a quick recap of this, and then we go back to the question. So just a recap of the mark scheme. This only involves photosystem 1. Light energy absorbed by the accessory pigments and pass on to chlorophyll A. Then electrons excited to a higher energy level, then collected by the electron acceptor, the primary electron acceptor, then passed along the ETC, then ATP produced by chemiosmosis, and electron returns to the PS1 or the chlorophyll. So this was actually for 4 marks, but you have a lot of about 8 mark scheme points, so any 4 of these would have been correct. Now I'll finish this video here and then I'll continue in the next video. So I've finished up to question number 3, and the rest of the questions will go on to the next video. Thank you everybody.